So what I wanted to, so I'm the product manager at Horizon Diagnostics, and what we set this webinar up to really tackle today is looking at how next generation sequencing can be brought into clinical laboratories, particularly on the validation side as well as the regulatory side. And so I'm going to give a very brief overview of where we see NGS sitting in the clinical space, and then my colleague Hannah, our product quality manager, will take over and then really delve into the details of how you can bring NGS into your lab, and the considerations you need to look at both the regulatory side of things as well as requirements and documentation and quality management systems. So I'm sure you're all very familiar with the big advantage of next generation sequencing, and that is that if you take a tumor sample, extract the DNA out of it, and then run it through your platform, whether it's IronTorrent or MySQL or another platform, you then have the ability to test or analyze the mutation status of a whole variety of genes. So taking, say, AKT1, BRAF, EGFR, KRAS, NRAS, and even in looking at KRAS, you could look at specific hotspots. You could go and look at the mutation status of, say, codon 12, codon 13, 59, 61, 117, 146, all from one sample, and that gives diagnostic labs great power and opportunity to gain so much information from just one sample. And potentially, as we move into the future, where I've put an FFPE sample here, it will be expected that this will actually be maybe a blood sample where you're looking at cell-free DNA. And those samples are going to be, you're not going to have much DNA, but if you can actually look at this whole range of genes and mutations from one sample, it's going to be a great advantage moving forward. Now, what's interesting, that's also very reassuring, is so this is data from a survey from Frost and Sullivan, and it's looking at what the key driver will be for NGS as a technology. And normally with any of these surveys, number one and number two will be consumables price or instrument price, and then they normally fight out the number one or number two spot. But what's really nice to see, and it highlights the importance of the data and the accuracy of NGS platforms and workflows, in that almost half the users are actually picking NGS on the accuracy they can achieve with the platform. If you then take that further, and this is some nice data from the Genome in a Bottle consortium that Horizon Diagnostics attends the meetings twice a year, is if you're looking for accuracy, so specificity and sensitivity of a workflow, here is a diagram where just one sample has been analyzed on 3 different platforms, and there's actually only a concordance of about of 80%. And outside of that, each platform will actually detect or not detect specific SNPs. If you then take that one step further, so that's the whole workflow. If you were to take then just one of those platforms and then analyze the data on 5 different bioinformatic workflows, you then only get a concordance of about 60% across those 5 different bioinformatic workflows. And so it highlights that not only is there a real challenge to understand where there may be either false negatives or false positives in the particular workflow, but also once you've actually run the sequencer, trying to make sense and clarity of the varying calling, it's a real challenge depending on the different tools you take to do that. And so if you take the workflow from start to finish, what you're looking at is you have your tumor sample, where historically it's FFP. It may turn to be in a blood sample. You have your tumor sample, and then it goes through to DNA extraction. You then quantify the DNA. go through library preparation. You have the sequencing on the various platforms you're taking forward. You'll then go through alignment and mapping, followed by variant calling, confidence scoring, analysis, and whichever action you take. If you look at each step, there's multiple chances of variability actually creeping in. For example, if your DNA quantification under-quantifies what you're looking at, then you would actually, if it over-quantifies what you're looking at, you'd then potentially actually underload into the library prep, and so you have less DNA than you would expect. Or the reverse could be true, where if you under-quantify, you overload, and potentially you could increase false positives through the workflow. And what this is all crying out for, and I think this is where well, what we've read and looked at through the New York State guidelines, is every single step is crying out for reference tools to be able to monitor how each step is performing, to give you an assay that you have full confidence in, that the actionable process at the end is taken with full assurance. I mean, that's my introduction over. What we'll do now is hand you over to Hannah, and then we'll bring about sort of more once Hannah's introduced the different regulatory space, how we're working in the regulatory space, or how you can work in the regulatory space. We'll then bring around to what we've developed to fit with, in some ways, fit with what New York State has recommended. And so, without further ado, I'll hand over to Hannah, who will delve into the details. Thank you.
Great, thank you, Jonathan. Hi everyone, I'm Hannah Murphy, Product Quality Manager at Horizon Discovery. Here at Horizon, I'm responsible for the quality and regulatory management aspects of Horizon's products, including design and new product introduction. Today I'm going to provide an overview of the regulatory requirements associated with the clinical application of next-generation sequencing, with a particular focus on validation. So in this space, there are a number of key regulators. CLIA is the first regulator. That stands for Clinical Laboratory Improvement Amendments. This is the body that regulates all clinical laboratory testing, with the exception of research performed on humans in the United States. The accreditation program under CLIA ensures the quality of laboratory testing and forms part of the reimbursement pathway. The second regulator is the New York State Department of Health. They provide CLEP as an accreditation program. This goes above and beyond the CLIA requirements, meaning most of the New York State is exempt from the CLIA requirements. This is a great source of best practice information, including the requirements for next-generation sequencing. The FDA, or Food and Drug Administration, generally regulate manufacturers of diagnostic tests in this context. However, recently published guidance documents a move towards regulation of laboratory-developed tests. Finally, proficiency schemes provide clinical samples to assess the variability of clinical testing. There are various schemes worldwide that help drive the quality of clinical testing outcomes worldwide. Finally, ISO 15189 is an international standard that is becoming widely adopted and combines general quality management system requirements with laboratory competence requirements. So what are the key requirements? So this is a combination of the requirements under CLIA, ISO 15189, and the New York State Department of Health guidelines. While there are a lot of similarities, there are a number of differences that exist between the requirements. So first of all, there are a lot of requirements for quality assurance, which aims to ensure the consistency and quality of delivery. Actions under the quality assurance include the generation of standard operating procedures and the retention of records. Secondly, validation requirements include general validation requirements as well as some specific applications for next-generation sequencing. Thirdly, data includes a vast array of information, not just the data obtained from sequencing itself, but data used to establish performance parameters from accuracy to sensitivity. These are key to next-generation sequencing, as a variation in primer or assay design could change sensitivity parameters such as the limit of detection. Fourth, quality control includes the control over samples such as pooling, as well as software used for next-generation sequencing. Good use of reference materials can help assist in the performance monitoring and determining day-to-day accuracy of equipment and operators. There are various report requirements, including the need to provide statements on limitations and incidental findings relating to next-generation sequencing. With new FDA provisions for laboratory-developed tests, there will likely be a need to provide the status of the approval of such tests. For this talk, I'm going to focus on the validation, quality control, and data aspects, although further information can be found on our website in the application section. Validation occurs across multiple areas of the clinical laboratory. Validation can include equipment, reagents, operations, and platforms. Two of the areas are test and platform validation. Test development includes establishing the protocols, optimizing performance, determining pooling parameters for patient samples, and using synthetic variants to compare tools and facilitate optimization. Test validation includes determining the parameters. The first test develops the highest validation requirements, and changes to tests must follow revalidation required against the existing tests to confirm comparability. Finally, platform validation includes cumulative performance data, determining the confidence intervals, tracking and validating different software versions, and also changes to platform must follow revalidation once again against the existing platform. Here is a summary of the headline next-generation sequencing requirements. On top of the general validation required by CLIA and other areas, these are the headline requirements under the New York State Department of Health guidelines. Laboratory-developed tests or laboratory-adapted tests require full validation. Commercial materials must be validated, whereas there is no clinical intended use. Performance characteristics of each sample type must be established, for example, FFPE. Vigorous validation removes the requirement to confirm every time a validation target is tested. All analysis software must be validated. Bioinformatics validity must be demonstrated. So there's a whole range of different requirements specific to next-generation sequencing for best clinical practice. So here's an overview of the data requirements. So the first point is the accuracy. So that involves the use of good reference materials. Robustness, which is the success and reproducibility of techniques. Precision, which is the analysis of which run sensitivity controls are used. Reproducibility and repeatability, which is analysis of between run, between operator, and even between different operating day controls. And sensitivity, which is around establishing the limit of detection. So there are a number of quality control requirements. There is a need for a no template control, which is included in all amplification steps, a negative control, which should be used for initial validation and periodically thereafter, and a positive or sensitivity control for every sequencing run. So this control should be a low positive DNA sample containing multiple known variants or mutations at challenging sensitivities to test the workflow and platform. A defined rotation schedule should be employed if not all variants can be covered in a single control sample. So in conclusion, there are requirements for all regulators that overlap. Validation is one of the most critical requirements to be considered, and use of reference materials can assist in this, but there is a clear focus on cell DNA over plasmid DNA. The future of FDA regulation of laboratory-developed tests is currently being developed, and the impact of this regulation remains up for debate. For further information, you can find information on the application section of our website, and the link is provided there. Thank you very much.
Thank you very much, Hannah. And so what we'll do is we'll bring the webinar round sort of into a circle back to a little bit on what Horizon Diagnostics has been developing in the lab to really work through and support next generation sequencing labs or molecular diagnostic labs to bring next generation sequencing into their workflow. And one question that I always ask people when I'm either talking to them or giving presentations is really delve into what is the impact of assay failure in your laboratory. And the answer varies depending on the laboratory. And this is where we really feel that Horizon can step in, because we feel that depending on if you do or you don't monitor the presence of, say, false positives or false negatives, which will always occur in molecular assays at a given frequency. What we want to do is support you to understand what the frequency may be of a false negative or false positive, so you have full confidence and assurance that the workflow you've adopted is giving you actionable results that you can trust. And so coming back to the workflow that I outlined previously, you've got the start, you've got your sample, DNA extraction, quantification, your library prep, sequencing, alignment mapping through to variant calling and analysis. Like I mentioned, there's a number of possibilities of variability and potentially errors creeping in, as you saw with the first few data slides I put together. And that's where the reference materials come in. We are working this space with various consortiums like Genome in a Bottle. We will be attending the FDA Next Generation Sequencing Workshop that is next Wednesday and Thursday, which I actually would recommend, and maybe we can put it in our sort of the follow-up email from this of the online login, because it's open. It's an open login for kind of off-site people to register and view, which I would, if you're interested in what we've presented today, then I'm sure that will be of interest to as well for you. And so when you sort of take a look at what Horizon Diagnostics is doing in this space, we always fall back on the core technology that we have at Horizon Discovery, which is our parent company, and that is gene editing capabilities. So what we do is we take wild-type cell lines from cell banks like ATCC. We put them through a single cell dilution to ensure that they're fully clonal. They then get put through pre-engineering cell line validation. So you have SNP6 to ensure that the cell line is what it is, and then we look at the loci using Sanger sequencing and digital PCR as well as RT-PCR to look at gene expression. And then when we're confident that we have a fully characterized wild-type cell or cell line, we then put that through our gene editing platform to then develop a clonal mutant cell line. And so, for example, we could take a wild-type SW48 and generate a NRAS A146T mutant. And what's nice and critical for the development of reference standards is the mutant cell line is identical to the wild-type cell line, except for that one mutation that we've incorporated. And so you can do direct comparators between the wild-type cell line and the mutant cell line, and also generate very nice limit dissection curves from the DNA extracted from these cell lines as required. And these go through the same extensive cell line validation step. And through this process, we've developed a whole host of, I think it's about 550 different cell lines, and here's a snapshot of the mutations that are most interesting in the companion diagnostic arena, where you can see we've got the BRAF, EGFR, the EGFR variant is actually expanding because we have the insertion coming through very soon. We have KRAS and NRAS, and also we're expanding those to include codon 13 as well as 117, and also the 59 mutations, codon 59 mutations. But coming back to next generation sequencing, what we've done for laboratories looking to both validate the assay or the platform initially, as well as using samples as a positive control moving forward, is we've taken a subset of our reference standards. When I say a subset, it covers 40 different mutations within EGFR, KRAS, BRAF, NRAS, PI3 kinase, and other biomarkers. They've had full quantification by digital PCR, so we know they're at 50% allele frequency. We've then used those to generate 4 different buckets, and we've called these C blends. And each bucket has 10 mutations, all at an allelic frequency of 5%, where each bucket has different mutations. And this fits in with exactly what the New York State really recommends in having standards for rotational standards or rotational positive controls, because you could imagine that you can take blend number 1 on this week to use routinely this week for every single run. Then next, in the second week, you can then move on to the blend 2, and then blend 3 on the third week, and blend 4 on the fourth week. And then next month, you then follow through on the same cycle. So you're always testing within a month 40 different mutations, and so you're showing that your platform can detect different mutations on different genes, as well as also mutations on different chromosomes. And we do cover SNPs as well as deletions. We will be moving into adding insertions as well as other types of mutations, more structural mutations. And so we're expanding the offering all the time. What's the other function of the standards we've developed is you can actually use these for establishing the limit of detection of your assay as well. So if you could take all of the C blends now, analyze or calculate if you can detect all 40 mutations at 5%. We've then taken blend 1 and 2 to generate a B blend that contains 20 mutations, all at 2.5%. Likewise, excuse me. Likewise, we've combined blend 3 and 4 to generate the B blend, and so again another 20 mutations at 2.5%. And you can use these either to establish a limit detection for validation, or if your assay is actually routinely has a limit detection down to 2.5%, you could use these as your rotational positive control. If you're actually trying to establish your limit detection even lower than 2.5%, we've then mixed these two blends to generate a one blend. So it's one tube has 40 mutations, all at 1.3%. And if you're moving into the cell-free DNA space, you could actually then take one of our characterized wild types and start diluting that to an even lower allelic frequency, or a lower copies per microliter, to really test the sensitivity and specificity of your platform. The next slide here is a snapshot of some of the data that you may be able to you may see if you're running a limit of detection using this panel of reference standards. And so what you have here is it's a subset of the mutations we have in the blends. So you've got BRAF, EGFR, KIT, KRAS, NRAS. We do all of our QC with digital PCR, and so this is the digital PCR result where we're expecting in the region of 4% to 5%. We then we've sent this sample to a partner, and they're able to detect these mutations down at that level. We've then sent them the 2.5%, and they've looked at the same mutations, and again they detect the mutations as expected. And then when you hit them with the 1.3%, which is it's a challenging sample, and I don't think next generation sequencing is really down to that level yet, but it's always good to push these platforms to their limits. And you can see that the lab started to struggle where you've got some dropouts of some of the mutations down at the 1% level. And so you can use this. Coming back to this, you can use this whole panel for either validating your platform to start with, or using either the 2.5% or the 5% as a positive rotational control in the workflow. What's coming next? We always like to tell people what's coming next. So we are working in the cell-free DNA space, so we're looking at very low copy number reference standards. We're also looking at translocation, so we'll be moving into the RNA world for there, probably with as a FFP format, and there's always lots and lots going on at Horizon, so there's many more other bits and pieces going on. That sort of brings me around to the last slide. Where we fit in, or where we want to support the labs, is what we do is, I mean, all that Horizon Diagnostics does is manufacture reference standards. And so if you are running any validation, or you need a reference standard for routine use, come and talk to us, because if we don't have it already, then we can certainly look at ways to develop that with you. And we work with molecular labs, work with next generation sequencing labs. Any labs, if you are already running KRAS or BRAF assays using maybe Sanger sequencing or qPCR assays, what's really nice with the reference standards we've developed is you can do complete bridging. You can do bridging studies because it's a renewable source of reference standards. So you can see how the reference standards perform in your current assay, and then do the bridging study on the new assay. And with the with the expectation, it'll show the new assay is actually more sensitive and more specific, as well as giving you the full breadth of mutation coverage. And if you are either working in assay development or platform development, we are open to OEM partnerships as well.